early onset mutations in APP, presenilin 1 and presenilin 2 genes. These we cannot do anything. What we get from our parents and when we age. On the other hand, right hand side, modifiable factors. We can control, we can change with our daily life, what we do, exercise, diet, and maintain your cardiovascular functions, and hypertension, diabetes, obesity, and metabolic syndrome, smoking, drinking alcohol, depression, traumatic brain injury, and cognitive reserve. The right hand side on the slide here, we can actually control, we can maintain. All these factors may contribute, will contribute to the dementia. If we can take care of these things, we can have a graceful later life or in our life. So this is the genetics of Alzheimer's disease slide. It is a very typical student version. I just want to mention one or two things. If you look at the amyloid beta, presenilin 1, presenilin 2, and these three genetic mutations in these three genes cause amyloid beta levels overall. Particularly amyloid presenilin 1, presenilin 2 increases toxic form of A beta, that is a peptide, longer form of A beta in the brains of Alzheimer's patients, familial. Whereas ApoE4 genotype, a sortilin related receptor protein, this increases overall A beta levels. On the other hand, the clusterin and complement component receptor 1 polymorphisms in these two alleles, they may not be able to clear the amyloid beta from the brain like a normal cell, normal neuron. So the clearing mechanism is defective in these two loci. There may be other loci as well, involved within similar way. But amyloid beta is being produced all the time in neurons, but they are being cleared. But in the polymorphisms in these two loci may not be able to clear. That's why they accumulate over the period of time. Now, this slide shows the cellular pathological changes in Alzheimer's disease. You can see some major ones I listed here, including mitochondrial fragmentation, loss of synapses, as I mentioned earlier, amyloid deposition, the toxic protein called amyloid beta, and they initially they are soluble and they become insoluble, and then they form diffusible plaques and matured plaques. All this form of amyloid beta are toxic, and also microglial activation, in other words, inflammation in the brain, and phosphorylation of tau, neurofibrillary tangles. All the changes, what you see here, including cell cycle deregulation, hormone imbalance, mainly in women, and all these changes may lead to neuronal loss and ultimately dementia in elderly individuals. At the therapeutic point of view, strategies have been developed based on mainly these cellular changes, what you see here, and currently being tested in animal models, particularly mouse models, and some molecules are being tested in the clinical trials in MCI, mild cognitive impairment patients, and Alzheimer's patients. However, unfortunately, I have to tell, we do not have any single molecule or drug that can definitely delay or prevent the progression of the disease. Answer is no. That's the current status. Now, this is the slide I was mentioning about earlier, amyloid beta formation in Alzheimer's patients. If you look at the left side of the slide, like this is a pathway happens in healthy, non-demented individuals. In the right hand side, Alzheimer's patients, mild cognitive impairment cases, see the red brick here that has been chopped right in the middle and forms the soluble APP alpha and the right side C83 fragment. Because of the presence of the alpha secretase enzyme in healthy individuals in healthy neurons, this brick has been further chopped, they become smaller, and these small fragments of amyloid beta are being cleared from the neuron every single day. On the contrary, in Alzheimer's patients, this brick, there is no alpha secretase. and 1 to 40 amino acid residues brick is being accumulated over a period of time, and that causes subcellular dysfunction in the neuron that may lead to several cellular damages. One is synaptic damage, which causing microglial activation, and also tau phosphorylation more and more. The brick amyloid beta 1 to 40 or 1 to 42 happens in Alzheimer's patients' neurons. That's not good. We also proved in the laboratory using the Alzheimer's mouse brain tissue. This is the 4 kilodalton amyloid beta, the toxic, and that forms, the brick is very sticky. It sticks to each other and forms much bigger forms of toxic protein, 16 kilodalton, 30 and 50, and full length amyloid beta is 110 kilodalton. This is mainly represents the size of the protein, toxic protein accumulation. This slide is an example of how amyloid beta are being accumulated in the form of soluble or even the insoluble deposits. So the top panel shows the 12-month-old amyloid beta mouse section, brain section, and the bottom one, 24-month-old. You see the difference? So much amyloid beta deposits accumulation over a period of time. These amyloid deposit formation, accumulation is purely age dependent. That's what we know. Alzheimer's disease is aging disease. Until 75, 80, we are all fine. We do our functions every day, and after that, we may not be able to do our own things. There we need our support and things like that. This is mainly due to the accumulation of this toxic protein in the brain. And there is another pathological hallmark I mentioned earlier called phosphorylated tau. If you look at the top panel of the figure here, in healthy neuron, the tau is bind to the black bar here. That's called microtubule, and allows all the small cargoes here, the blue, the red one, mitochondria, they travel from the center of the neuron to the terminal. This is the terminal called synapse, a presynaptic terminal, and connecting to postsynaptic terminal. That's another neuron here. That's the way the neurons communicate. And where the red one here called mitochondrion supply energy for all these functions. I'll show you much better later. The other cargoes, the blue and green, these are very important for all the synaptic functions. Whereas you look at Alzheimer's disease neuron, the tau, the clip here, become phosphorylated and detached away from the black bar. So when the phosphorylated tau is not bind to the black bar, the cargoes, mitochondria, and other subcellular organelles may not be able to move from the center of the neuron to the terminals. Look at the numbers, just half of the healthy neuron numbers here. This is the synapse. This is basically the defective synapse, and they may not be able to do the job. And eventually, if a number of mitochondria and other cargoes are lesser in number, the synapse becomes degenerate and the neuron die. That's what exactly happening in Alzheimer's disease patients, or even mild cognitive impairments. So what we need here, we need more mitochondria, more other subcellular organelles, the one in the blue, the one in the green here. Then the neurotransmission can happen a normal way, and we can restore our cognitive functions. So this is another pathological hallmark called inflammation. What you see in the middle red amyloid deposit surrounded by tiny, tiny cells called microglia. Why they are here? Because they wanted to swallow the insoluble deposit, but they may not be able to do that. This is sticky, strong, very difficult to dissolve. So they don't give up. They try to produce more and more. The more glial cells or even astrocytes cause the inflammation. That's another pathological hallmark in Alzheimer's disease. And then last one is mitochondria. It's my favorite. Look at the left side, small number, intact, healthy, structurally intact mitochondria. Okay, this is cristae, and where the electron transport chain is localized where it's, the mitochondria produce ATP, energy to the cell. So whereas right hand side in Alzheimer's neurons, you look at them more in number, but they're almost like a swollen potatoes. There is no cristae like here. This is the healthy, actively functionally active mitochondria. Whereas here there is no cristae, they don't produce any ATP, rather more in number. These are structurally damaged mitochondria, and these are the things present in Alzheimer's disease neuron. Now, I'm going to say one or two sentences about oxidative stress, oxidative damage, and mitochondrial dysfunction. It may be too much for you guys, but in a simple way, if you look at the healthy cell, reactive oxygen species, in other words, toxic oxidants production and antioxidant defense enzymes. In a healthy cell, these are 50-50. The toxic oxidants production and antioxidant enzymes presence endogenous, they are almost equal. So that means the cell is able to survive, do the job, produce ATP, and maintain normal functions. Whereas an Alzheimer's cell, such as like this here, antioxidant enzymes are present, but the free radicals, in other words, toxic oxidants are higher in number. That's what it is imbalanced. That's what it is called oxidative stress, oxidative damage. That's what happens in Alzheimer's disease neurons. Not only in Alzheimer's disease neurons, but also in, in aging condition and Parkinson's disease and ALS, Huntington's disease, several human illnesses. So the oxidative stress, mitochondrial dysfunction is a common, abnormality in cell. And so what are these mitochondrial, what are the changes in the mitochondria? So I just listed some here, maybe too much for you guys, but I just read increased production of oxidants and reduced mitochondrial membrane potential and enzymatic activities. Mitochondrial DNA changes occurs in aging, okay? And also mitochondrial structural changes in transport and reduced mitochondrial biogenesis. We need mitochondria, new mitochondria all the time. The mitochondria may not live more than 2 to 3 weeks. That means they die, they recycle. The fresh mitochondria, new mitochondria is always necessary to maintain the neuronal function. Now this is a simple slide, what exactly mitochondria look like in electron and in ultrastructural level. The double layered membrane structures, and they produce ATP, they maintain calcium levels, and they also source of oxidants I mentioned earlier. And the oxidants are being scavenged by antioxidant enzymes, and the mitochondria critical, they perform apoptotic cell death. When cell is about to die, they just say promote the cell death, make sure the debris is not stuck in the cell, and also responsible for activation of caspase family of proteases. So this slide is a little bit too much, but I can explain. Mitochondria are synthesized right here, heart of the neuron, and then all the tiny ones here you can see, those are mitochondria. They travel from center to all the way to the terminals called synapse. And what they do? They supply ATP to several synaptic functions: vesicle fusion, neurotransmitter release, neurite outgrowth, synapse formation. All these functions are highly ATP dependent, are highly energy dependent. This is in normal healthy synapse. The synaptic for all synaptic functions, ATP is very critical. But if you do not have a healthy active mitochondria at synapses, all the functions what I mentioned here, they are not going to be normal. That's what synapse start dying. That's what we lose our memory functions. That's why at the bottom I wrote, keep your mitochondria and synapses healthy and active all our lives until we die. If we lose your mitochondria, if you lose your healthy synapse, we are going to get a memory loss and cognitive impairments. So this slide is to show what is the update on antioxidants intake I did a survey on mouse models and elderly individuals, and also Alzheimer's disease patients. And some of the antioxidants I listed here: vitamin C, vitamin E, and also combination of vitamin C and E, and several others: ginkgo biloba, melatonin, curcumin, the yellow spice Indian Asian community use every day in their meal, and coenzyme Q10, lipoic acid, many others. The bottom, several molecules, mitochondria targeted molecules. These target the mitochondria, and then they effectively work and maintain the mitochondrial function and structure. This is the summary slide from animal models of Alzheimer's disease. You can see there are 23 studies conducted in several lines of Alzheimer's disease mice, and you can see several molecules, starting from ginkgo biloba, vitamin E, melatonin, coenzyme Q10, and so many here. So out of 23 studies, 21 studies showed a positive effect, beneficial effects in Alzheimer's disease mouse model. How they did that? They reduced amyloid beta toxic protein, number one. They extend the lifespan, and also they improve the cognitive functions in the mouse model. Next slide is to show antioxidant use in elderly individuals. We call it as a perspective studies. So out of 12 studies, 8 of them showed a positive effects. That means improved cognitive functions. These are the longitudinal studies, anywhere from 2 years to almost 30 years. This is what tells this this antioxidant use. If you use these antioxidants early in life, you can delay the progression of the disease. That's very clear message. I'll show you another slide, randomized clinical trials.